Everybody. Hi everyone. My name is Stefan Erhardt. I'm faculty in the track. I'll just set the stage for just a minute. This is one of the trials that we did here in the Center for Clinical Trials. So about 2 years ago, we were contacted by the study PI, who in the beginning was asking questions about study designs and how he wanted to test convalescent plasma for to treat patients with COVID-19. And recall that was an emergency situation about 2 years ago, and we had many discussions, and these morphed into basically us taking on the role of the data coordinating center for a larger multi-center trial that we are going to review today. And us, that means Shariza, Dave, and I, together with our colleague Brian Lau. And there was also a clinical coordinating center out of the School of Medicine that was formed from a well-known group, and we had never worked with that group, so that was an interesting experience. But they were good. They interestingly brought the database. It was a commercial system. Word on the street was it could start up very quickly. And so we used that data system that we also didn't really know. And then we were looking for a funder, and basically David Sullivan, the PI of this, was successful in getting funding from the Department of Defense for this. So that was the main funder. A few NIH institutes pitched in some money. There was money from Bloomberg Philanthropies and also from the state of Maryland. This was really a trial that was set up very, very quickly. I want to say within a few months, we started this operation, and again in an emergency situation. So the worry was from the get-go in those clinical centers that were swamped with patients. We received terrible news from New York, for example, where people were lying on the aisles, and the question for us was always, at the end of the day, a clinical trial is a data collection operation, right? So would we be able, under such circumstances, to actually collect meaningful and valuable data? So that's a little bit of the backdrop of this specific trial. Okay, and I think Jasmine and Edward, please take it away.
Great, thank you. Edward, do you want to share your screen? Great, thank you. So good afternoon, everyone. So today, me and Edward, along with Dr. Erhardt, will be presenting a paper on the early outpatient treatment for COVID-19 with convalescent plasma. Next slide, please. Yes, so this is the article for today. And then, so some background for this study is that in the United States, 8% are hospitalized after getting infected with COVID-19. So for right now, most of the therapies are mainly targeting disease progression or death prevention among hospitalized patients who usually have more severe symptoms. But there's also a growing need for outpatients who have symptoms that are not severe enough to be hospitalized, especially like Dr. Erhardt said, when healthcare resources are tight. So the FDA issued emergency use approval for 3 monoclonal antibody therapies, for which of the data for these therapies showed decreased incidence in disease progression within 5 to 7 days after onset of COVID-19. But however, we'll need alternative outpatient therapies when monoclonal antibody therapies are unavailable, limited, or ineffective. So for the convalescent plasma, they can be obtained from donors who are infected and have recovered from COVID-19. There are previous studies done that suggesting no safety concern are found among those who are hospitalized, so suggesting that that might be okay to use. And then there are also improved outcomes associated with provision of high titer plasma within days of symptoms onset. However, there are not enough randomized control trials done with outpatients. So a similar trial was done in Argentina that showed reduced relative risk in disease progression, but unfortunately, other trials halted due to fertility. So for this study, the study objective is to see whether transfusion of COVID-19 convalescent plasma within 9 days of symptoms onset will be effective in preventing disease progression, leading to a patient being hospitalized. So yes, for the methods part, is a double-blinded randomized controlled trial. Although it is double-blinded, but actually all participants, providers, and statisticians were masked in the trial. And there are 2 study arms. So one with the plasma from qualified recovered COVID-19 patients, and another one being the control plasma. And the plasma was collected at the blood banks across 23 sites in the United States. And the general idea is to basically assign COVID-19 positive patients within 8 days after their symptoms onset to a study arm, whether that be the qualified plasma or the control plasma, and then have the transfusion by day 9. And then as for the participants, some of the features of the study population includes those at least 18 years old, regardless of coexisting conditions and vaccination status. So as a reminder, this trial recruited from June of 2020 to October 2021. And then the majority of the public did not receive their first dose until April of 2021. So for most of the recruitment period, most people were not vaccinated. They also included pregnant women in the study, and then managed to enroll over 1,000 participants during the relatively early stage of the pandemic, where a lot of things were uncertain and unknown compared to now. And then, so the first question I have for the audience, that given this high number of participants enrolled, what could be some of the recruitment methods you can think that, or read in the paper, that helped achieve such high number under emergency situations? Feel free to use the chat too.
I think one thing to consider is that in terms of in-person interactions with potential participants, that was pretty much non-existent. So relying on electronic sources was pretty critical.
Yeah, that's a good point. Yeah, so next slide, please. Yeah, so what they did in this study is that they recruited through a lot of sources, I would say, such as clinic-based lists of positive outpatients, physician referrals, participant referrals. And then a very interesting point is to look at is that they actually did quite a lot on media outreach. So paid media ads were posted near clinical trial sites, where they directed interested participants to a call center or a website. Like Teresa said, like a lot of the in-person interaction was not possible during that time. And then so staff will then confirm if participants could be treated safely on an outpatient basis. And then they also have earned media stories, have ads posted on search engines, social media, high volume COVID testing sites, local media outlets, etc. So we can see that actually this trial made use of the media for recruitment, and then hence getting a high number of enrollment in a relatively short period of time. So this actually ties in with the last journal club, where Kamini and Daisy present a very interesting article on what kind of role can social media play in clinical trials. So I thought that was interesting.
Yes, and I can add a little bit of color here. So we worked together with a company that was, I think, very good, very savvy, and very expensive. And the company did a lot of this media outreach, and that was, you know, investigators talking on the radio. There were advertisements. There were a lot of things happening. So this company was very good at bringing in people and at you know, activating people to participate in clinical trials under these emergency circumstances. Jasmine already mentioned call centers that you could call. These call centers then pre-screened people, and then would then say, you should go to this or that clinic, so to the next clinical center where another layer of screening happened, and then finally the enrollment. And this was also a challenge for us, because the data that were collected by these call centers, by these companies, they basically did their thing. But we saw relatively quickly that those data were very hard for us to work with and to understand, because what they thought was screening for them to bring people into the trial was not really the data that we needed to populate our concept statement to begin with. So merging those different data sources, and some of these data again were created not really for trials purposes, was one of the challenges that we faced, I think, early on.
Yeah, I also imagine that whoever was reached by media, usually they're relatively younger compared to older people too.
Right, and I think we see that, right?
Yes.
Did anyone else get advertisements on their Instagram or social media pages?
I do.
Did you get this one for this study?
No. I got for other trials, but yeah. Next slide, please. Yeah, so and then for the exclusion criteria, it includes previous COVID-related hospitalization or planned hospitalization within 24 hours after enrollment, and also for those who have reactions to blood product transfusions or previously received monoclonal antibodies, and also obviously if they cannot adhere to protocol. So moving on to the randomization part, the trial did a 1-to-1 ratio by a web-based system with permuted block sequence. It's also stratified by trial site and participant age as well, so less than 65 and 65 or above. So the products were, both products were matched on blood type compatibility as well, and then the plasma was transfused for an hour within 24 hours of enrollment to the study. And then lastly, lab tests were also done to confirm the presence of antibodies in the donors, just to make sure their plasma qualifies. So here you see figure 1. We can see the flow chart where almost 6,000 participants were assessed for eligibility. That's a lot. And then most of them were excluded because they did not meet the inclusion criteria, and were left with 1,225 who were randomized into one of the study arms. However, you can see that in both groups, there were some that did not receive treatment at all. So therefore, they're excluded from the analysis. So for our primary outcome, it is the COVID-19 related hospitalization within 28 days after plasma transfusion. That is assessed and compared as the cumulative incidence between the two treatment groups. So death before hospitalization was specified as one of the primary outcome in the protocol. Unfortunately, that did not happen in the trial, so the outcome is equivalent to the COVID-19 hospitalization. So moving on to the analysis plan, in order to detect for a difference of at least 25% with 80% power, a sample size of 1,280 is needed. But because to account for potential loss to follow-up, the final planned sample size was increased to 1,344, and then the actual enrollment was 1,225, so that was really close. And then, so a second question for the audience is that what analysis do you think they used in the study, and what are some of the features for this type of analysis?
Jasmine, are you referring to the power calculation or the actual primary analysis?
The analysis. Yeah, I saw Ling's chat. Yes, that's correct. It's a modified intention to treat analysis. Next slide, please. Oh, one before that. Yeah. So they used a modified intention to treat analysis. So the traditional intention to treat analysis where you analyze according to the original assigned treatment, basically regardless of what happened later. But in this modified intention to treat analysis, they excluded participants who did not receive any transfusion of plasma. So those are the 18 from the convalescent plasma group and another 26 from the control group. So with this analysis, they also calculated the risk difference and also the mean survival time, which is the expected mean time to hospitalization restricted to 28 days. So as previously mentioned, the primary outcome is assessed by the cumulative incidence in the two groups. So baseline variables are also adjusted to increase precision of estimates and also account for any potential dependent censoring. So these variables included age, vaccination status, time between patients' COVID exposure, and plasma transfusion, etc. And then I'll hand it over to Edward.
All right. So I'll be going through the results and the discussion part of this article. So looking at table 1, the baseline characteristics between the convalescent plasma group and the control group, there is no obvious imbalance as far as these different treatment groups in terms of age, female sex, BMI, and also other coexisting conditions or vaccination status. And they actually listed some other variables, including some clinical vital status. And generally speaking, there's not much difference between the two groups. Sorry. So this figure is the primary outcome. They calculated the cumulative instance of COVID-related hospitalization. So on the left, it's the unadjusted cumulative instance and including a confidence interval. And it's pretty obvious that the convalescent plasma group has a lower cumulative instance to the hospitalization. On the left, on the right, it's the adjusted targeted minimum loss-based estimation model. And basically the results didn't change much between these two methods. So in this table, we can look at the first row, the primary outcome. So in convalescent plasma group, there's only 17 patients being hospitalized after the convalescent plasma treatment, and there were 37 patients in control plasma being hospitalized. And the relative risk reduction was 54%. But the absolute risk reduction is only 3.4%. And these differences are all statistically significant. And the authors also look at other severity index after hospitalization. And they found that within those hospitalized patients, there are 3 deaths in the control plasma group and no deaths in convalescent plasma group. And a bigger difference is found in the non-ICU hospitalization with supplemental oxygen, and meaning that those patients was severe enough to receive oxygen supplement, and there are more patients in the control plasma who needed supplemental oxygen. And the expected time free of hospitalization were pretty similar between the groups, and the probability of remaining free of hospitalization was also pretty similar between the group. Even though the p-value is pretty small here, but the pre-specified p-value was even smaller than the 0.004 or 0.006. And the authors did a lot of subgroup analysis. So they stratified by sex, BMI, age, vaccination status, and also some other biomarkers. So I think the most interesting part is that if we look at the vaccination status, we can find that over 80% of the patients enrolled in this study didn't receive any vaccination. I think part of the reason is that the vaccinations started to roll out. I think near the end of 2020, and at that time the trial had begun only for 3 or 4 months. So not a lot of people in this study received the vaccination, but we still see that among those either fully vaccinated or partially vaccinated, the convalescent plasma effect is more prominent. And also those receiving the convalescent plasma treatment or control treatment within 5 days, the effect is much bigger than those receiving the treatment after 6 days. And another thing to look at is the biomarker, specifically the CRP level and the absolute lymphocyte count. So we can find that for those with a normal level of the biomarkers, the effect of convalescent plasma is more prominent. All right. So the discussion session, the main finding is that the convalescent plasma can decrease the instance of hospitalization. And based on the absolute risk reduction, we can get the number needed to treat is 29.4. And the authors do.
Maybe can I ask, what do people think about this number needed to treat? Is that good or is that not so good?
Yeah, Dr. Earhart, I was very curious about this number because in some other trial we see like 5 or 6 or even 10. Sometimes we consider 10 would be a very effective treatment. I don't know. I'm very curious.
Mm-hmm. Yeah. So the number needed to treat, is, I think, is a nice metric. Has been criticized also by some, but it's a metric that gives us some idea how many people we need to treat in order to prevent one bad outcome. And a number needed to treat of 29 is not great. And typically a number below 5 is really good, and sometimes based on the condition, as you say, Edward, is 10. 29 is not great, but that's also due to the smaller number of events that we had, right? So it's dependent on these factors as well, not only on the treatment, but also on the number of events.
Thank you, Dr. Earhart. Yeah, so the authors also mentioned some of the.
Sorry, just a quick question. So Stefan, you had a low number of events in these groups, but how does that compare to what was the epidemiology? In other words, was this patient population somewhat healthier and that's why you had a low number of events? Or was this sort of consistent with the epidemiology that only 3% are, that only 7 or 8% are going to be hospitalized? And the reason I ask is that it influences your interpretation of the number needed to treat. If in fact out in the real world, 30% get hospitalized, then the relative risk reduction becomes pretty impressive. If in the real world only 5% get hospitalized, then using this a lot may not be brilliant. So I just wondered what you thought about that.
Yeah, Doug. See, thanks. Remember at the beginning we had no idea. The numbers were extreme. When we started this, the numbers were extremely imprecise, and we actually didn't know what to expect. But I think overall, looking back now, this is probably roughly what the epidemiology showed at that part of the trial. And that changed during the course of the trial also, because once the vaccines came in, the epidemiology changed. There were the different variants that came up that changed the epidemiology. I think we'll be talking about this one as well. So that was a very, very hard trial to power in the first place.
All right. Yeah. So like Dr. Elhart said, prior to this study, there are mixed results in terms of either convalescent plasma or monoclonal antibodies. The effectiveness is not very clear. And some of the reasons, including the lack of modern study designs, smaller sample sizes, and differential viral response to different kind of antibodies. But one thing that's clear is that the key for effectiveness for any kind of antibody therapy is that the early administration and sufficient amount of antibody would be very important for the efficacy. So now a question for the audience. So what are some of the differences between convalescent plasma and monoclonal antibodies? Because these two type of therapies are both used in COVID-19 treatment. Sorry. Yeah. Can anybody think of any difference, like the price or even the process of the production?
Yeah, Link says in the chat, polyclonal versus monoclonal.
Yeah.
With a rapidly changing virus too, the convalescent plasma is probably quicker to catch antibodies that are specific to the one that's locally circulating as well.
Yeah. And that reminds me, there was another dimension of the whole thing, because the thinking was that we tried to source the plasma locally. Right, so we did not have one big center and shipped centrally to all different places or study centers, but we tried to source locally because there were early indications that that the local composition of the strains were somehow specific and might be different across different cities and study centers. And I can't tell how true that was retrospectively or now in hindsight, but at the time it was deemed important to source locally.
Yeah, we did look at it. There was definitely a split west and east of the country, but I think any smaller than that, it's a little bit harder to determine.
Yeah, thank you. Those are very good points. So just very simple explanation. The difference between convalescent plasma and monoclonal antibody is that the source is a little bit different. We can get the donor blood to produce the convalescent plasma from recovered COVID-19 patients, but the production for monoclonal antibody is a little bit more complex. The activity, just like Lynn said in the comment, the convalescent plasma has polyclonal activity, and there's a more diverse mix of antibodies with different functions. And on the other hand, a monoclonal antibody is monoclonal, so it only has a singular predetermined target. For example, like the spike protein of SARS-CoV-2. And generally speaking, it's cheaper to produce convalescent plasma, and it's relatively expensive to produce monoclonal antibody. And like Dr. Bach said, that the convalescent plasma potentially can be more useful in a rapid change environment because it's less vulnerable to resistance. And the monoclonal antibody instead is probably more vulnerable to the antibody resistance strains. So some other considerations, like Dr. Earhart said, that in some countries, especially in low middle income countries, it's more difficult to get the monoclonal antibody available. So convalescent plasma could be another choice for them, and there's no patent limitations, and it's cheaper, so it's easier to get to the patients. And also, like the last point in these slides, there's potential utilization for a system that is built targeting the local variant that could be more specific than other kind of therapy. So the hypothesis of the monoclonal antibody or the convalescent plasma is that the most hospitalizations were due to the symptomatic hypoxia from pulmonary inflammation. And the plasma antibody has the function of direct neutralization of the virus. And also it can activate a complement system in the body, and also can facilitate the viral particle phagocytosis and all the other kind of cytotoxicity. So those things can suppress the immune system from reacting in a way that causes the patients to have severe symptoms and hospitalization.
And if I may chime in with one more point that just occurs to me, just when we were ready, or when we were coming close to publishing the results, the WHO came out with a with a statement that there was no place for convalescent plasma in the treatment of COVID-19, which was at the time, very. It was upsetting to many on the study team, and subsequently we were actually rejected in the first try at the New England Journal. Then we appealed, and then there was a long process until the paper was finally accepted. I think the logic of the WHO at the time was that especially in low-income countries, safe blood products are still difficult to come by, and they were worried that under these emergency situations, plasma was used without proper safeguard, perhaps in an environment where the HIV prevalence was very high, and the prevalence of other bloodborne diseases was high, et cetera, et cetera. So this was another layer of complexity. So there was steep headwind for convalescent plasma for quite some time.
So correct me, but I think as I remember, weren't there also studies in ICU patients that showed that convalescent plasma was ineffective in that group?
Yes, there were those studies, and there were concerns about the sample size. There were concerns, tough concerns actually, and I think warranted concerns about what kind of plasma are people getting. So is that really high titer plasma, or is that low titer plasma? So we really transfused high titer plasma, and many of those early studies did not titer their plasma, also because methods were not readily available. So some of these assays were developed on the fly. And then there were also different patient populations, different endpoints and all that. So the picture was very, very unclear.
I also think might want to comment on the logistical triumph, which was getting all the plasma. Blood banks got tons of convalescent plasma, and never did we seem to run short during this trial.
Yeah, very true.
Yep. So following on Dr. Earhart's comment, so this trial is conducted when the pandemic was just heating up with a lot of new variants. So can anybody think of some challenges that the investigators probably had to face I mean, other than the WHO and the journal publication side. So I will just go through some challenges mentioned in the article, and Dr. Elhart, feel free to jump in anytime. So throughout the trial period, because it's fast changing environment in terms of COVID-19 pandemic, so the standards of care and available therapies are all changing throughout the trial period, and many indications for therapies are also changing. Another thing is that the variants became more prevalent during the trial period, and specifically the Alpha variant and the Delta variant in the 2021 summer. And so because of these variants are new, but the plasma was obtained before these variants emerged. So we don't know if those convalescent plasma acquired before the variants could be used under the circumstances of new variants. And another thing is that the hospitalization in control group was not much, it's a little bit lower than the 8% in the United States. So that also is part of the challenge in terms of the representativeness of the population. And another point mentioned by the authors was the trial was probably not large enough for a definitive subgroup analysis. So, Dr. Elhart, do you have any other comments to add here?
No, nothing to add. And you're right. So with the subgroup analysis, some of these subgroups are small, but at the end of the day, you don't power your trial for subgroup analysis, right? So you typically you get what you can, and then you look into subgroups, and you see something or you don't, right? So it was, I think, overall, I think especially for that time frame and the circumstances, it was a pretty large trial.
So some of the strengths of this study is that it's a large and multi-center trial, so the population was pretty diverse, and the methodology was pretty rigorous, double-blind intervention with a controlled plasma. And also the percentage of patients adhere to the protocol was very high. And we also included the pregnant women in the study, which this population was very often ignored in all kinds of trials. And also this study has implication in resource-constrained areas, especially for countries that the monoclonal antibody is not available or the vaccination cannot be distributed very equally. And also based on this, the results of this trial, the future pandemics can be probably can be mitigated by more therapy options. All right. So that's all for our presentation, but we can certainly do some more discussion.
Yeah, I'd like to add that I was very impressed with all of the work that Hopkins did on this in both the School of Public Health and the hospital. But the biostatistics department contributed some methodology on restricted mean survival time. And I thought also, I was impressed that restricted mean survival time was actually used. We're beginning to see that now. It's a method that is preferred when the proportional hazards assumption of the Cox model doesn't apply. But even though we've had RMST for a decade at least, nobody's used it until recently. And now the biostatistics department, as you may have read, contributed some new methodology so that covariates could be used in RMST as they are in Cox's model, although not yet is well developed, but a beginning of it. So I think many departments were involved in this, and they all contributed excellent work.
I'd like to comment further on that because I completely agree, Jay, but interestingly, we had a very difficult time explaining restricted mean survival time to the clinicians, and there was frequent confusion and concern that it was not a valid methodological approach. I can't tell you how many times people questioned whether this was the appropriate way to present the results, even though it had been pre-specified. And things were moving so quickly that it just became a bit of a challenge.
It's really interesting, David.
Yeah.
I think as we speak, there are statisticians having a hard time explaining Cox's regression model to clinicians.
Sure.
But I think, yeah, that's interesting. I'm not surprised because it's something very new. What I see in oncology today is people will use the Cox's regression model with covariates, and then they will use, as they call a sensitivity analysis, the RMST. But the RMST does not include covariates, which is very nice and up to date. But what happens if the two methods disagree in conclusions? And then nobody knows what to do.
Absolutely. I thought if it's okay, I'd bring up a couple of other topics that were really kind of interesting challenges for this study. And one in particular was the pre-specified use of a modified intention to treat population, which took many, many back and forths arguing with the New England Journal because their position was that only an ITT analysis could be accepted. I don't think it led to the initial rejection, but it took a lot of arm twisting to get them to accept us reporting results in that modified population.
So why did you use the modified ITT as opposed to a straight ITT?
I'm not sure that I was part of the design part of that, but I believe that the rationale was pretty sound in that the duration between randomization and transfusion was thought to be very short, not likely to be related to anything that would have affected our analysis if someone didn't get transfusion. And there's no plausibility for a lack of effect or an effect in people who didn't get the transfusion. So some of the rationale for always preferring ITT didn't necessarily make sense in this particular study.
And what do both of you think about the subgroup analysis on time to transfusion from onset of symptoms? I mean, it really looks like you have a 3-day window and that's about it.
I think the clinicians here would argue for a 5-day window. That's why that breakpoint of 5 days is what's presented there. They would have postulated even before the study results were available that earlier in the course of the disease was far more likely to show a benefit. As Stefan noted, there were some trials who did not show any treatment effect, but those were patients that were much farther along in the disease, and that's consistent with that particular approach. We didn't have the power to prove conclusively that early versus late made that significant a difference. We're still looking at it.
Yeah, and at the time, I remember there was communication with people who had experience with convalescent plasma in the ICU setting, right? So people already on the ventilator, severely sick, and they argued that convalescent plasma had no effect. And at the time, our explanation was that this was likely much too late in the process.
Yeah, another really influential trial looked at patients in the emergency room. And again, that's also many people thought was too late to be likely to show an effect, and that their lack of effect really did influence a lot of concern about our trial, which was much earlier in the disease process for most people. I think another point, especially perhaps pertinent for students, is the statistical analysis plan for this trial pre-specified the use of a one-sided p-value. That was pre-specified, but nevertheless, that's another place where the journals were just extremely reluctant to accept it. And in fact, in this case, kind of compelled us to report a two-sided p-value, which thank goodness was also significant. And I think even that's why during the student presentation, they made a comment about the pre-specified being more relegated to a footnote.
Yeah, well, if the New England Journal rejected this paper, you might have considered rejecting the New England Journal. Because that is really ridiculous. And we know that the p-values are not all that meaningful anyway, a big controversy.
Yep.
One-sided at the 0.025 level is a good compromise, and you could have offered that, but I'm disappointed in the New England Journal and not in Johns Hopkins.
Well, thankfully here it didn't make any difference, and eventually it was published. Wouldn't have mattered. But I think for myself, I would probably not put forth a protocol that specified a one-sided p-value, just thinking it's not worth the effort. And the effort here was considerable.
So why wasn't a one-sided p-value proposed?
Why was it?
Yeah.
I presume as being protective against what they would have felt was an uninteresting finding that the convalescent plasma was worse than the control. Who cares? If it's not better, then no one is interested. In the context of an emergency pandemic, wanting to get quicker results, this is probably a power-saving approach.
Well, considering the fact that it, at least theoretically, could be worse, I'm not sure that I would have been a big fan of a one-sided p-value, because the results really could have gone the other way.
Yep, but like I said, I don't think I would do a one-sided p-value in a future trial.
Yeah, but I think the issue is it could have gone the other way, but is there interest in showing that the other direction is statistically significant?
Yeah.
Right. I think in the discussion of challenges to the study, I think it's also possible that one challenge was the overall politicization of the pandemic, but then the fact that the Trump administration allowed an emergency use for this product before there really was any data at all. And I think that really clouded a lot of people's minds about whether the trial was properly being performed. This trial was already underway when that emergency use authorization came out. I don't think it had enrolled patients yet, but it was already it had already gotten started.
I would just like to chime in. This paper I noticed is published relatively close, published March of this year, but all of this work is actually done before vaccination, when people settled down. I was just maybe Dr. Erhard, Dr. Shade, or Teresa, who's on the trial, might comment on this. Moving forward, how can research, especially clinical trials, how to adapt to rapidly evolving times like the pandemic right now? Because I'm pretty sure this is very discouraging for many people who like to work in trials for infectious disease, right?
Do you mean how rapidly the vaccine was developed and came out during the study?
Yeah, how can research world can adapt to that? Because I imagine, because running a clinical trial takes a long time planning, but then as you said, a lot of things change over the course of the trial, right?
Yeah. We could talk about some of the discussion we had when the vaccines came out and what to do with those individuals who were actually vaccinated. I mean, we had several options, right? I wasn't privy to these conversations, so Stefan and Dave, please chime in. But I have the rough idea of what had happened. Essentially, we could include them, or allow it after randomization, right? And so for this particular trial, we decided to include them, but we were also running a trial where we were looking at prophylaxis with the use of convalescent plasma, and we decided to exclude them there. There's certainly benefits and drawbacks to both approaches, but that was the final decision. Same thing with monoclonal antibodies too. And just to kind of pile on to that, at the time that we were trying to publish this paper, the monoclonal antibodies were showing a lack of efficacy against Omicron. So what did this mean for convalescent plasma as well? So we were also dealing with that particular political argument.
Yeah, and Jasmine, to your maybe a bigger picture answer, we felt like we were constantly surprised by changes, as lots of things were happening, and yeah, I think for the field of infectious diseases, when you have a disease where you have more events, then you are probably, power wise, you're more on the safer side and can do this more quickly. We were, I think, that with the relatively low number of events, we were forced to sample so many people into that trial, more than 1,000, and that would be different, or could be different in a different disease that's just more aggressive, Jasmine. So it really depends on the type of disease. It depends on the healthcare setting. The healthcare is very good here. It might be very different also in a different country, right? So, but here we felt that constantly things changed around us, and we needed to kind of make things happen.
Related, I think, I think the speed with which these vaccines were developed was so astounding and unexpected and not likely to come up in future trial considerations. And I sure hope we don't have another pandemic, which provides the substrate for such a rapid vaccine development. I actually myself thought that we should exclude anyone who had been vaccinated. I was very concerned about diluting our treatment effect, because we presume that people who had the vaccine were not going to get hospitalized. That's what the vaccine data showed. And I think we were fortunate that we still were able to demonstrate a treatment effect. Didn't have a ton of vaccinated people in the study. Here's another interesting methodological thing, at least interesting to me. The clinicians and the study PI were very, very interested in almost all the time described the treatment effect in terms of a relative risk reduction. The journal absolutely refused to highlight the relative risk reduction and instead focused specifically on the absolute risk reduction. And, of course, both of them are important. And it's interesting, I still find it fascinating the way people communicate results of things like this. We talked here about the number needed to treat, which is really more focusing on the absolute risk reduction. But still the clinicians often will talk about the relative risk reduction. And it's heightened my awareness anytime I hear now a relative effect in any context having nothing to do with plasma. I can't help but remember all the discussions that we've had here about it.
Well, so I'd like to speak up for the clinicians, because not all clinicians believe that it's only relative risk. Maybe in this trial they did.
Yeah, I don't mean to categorize a big group, sorry.
Yeah, but what I would point out is that I think both are important.
Yeah.
Because it's like the old meta-analyses of oral corticosteroids and peptic ulcer disease. And one showed none, and the other showed that there was a 50% increase in the risk from 1 to 1.5%. And the meta-analysis had, 10,000 patients in it. And it says, okay, it is statistically significant and biologically not meaningful. So I think you need both to understand the importance of the data.
Absolutely.
So I'm a little bit curious about the clinical utilization of the convalescent plasma in outpatient settings, because there would be much more outpatient patients compared to hospitalization patients. And how do we treat those patients in a real world setting? Do we give all of them convalescent plasma if those are available, or we just target some kind of high risk patients?
So, I'm by no means an expert in what happens in COVID-19, but my understanding is that once you're in the hospital, there's little evidence for efficacy of convalescent plasma. And there's not much evidence for efficacy of prophylaxis, preventing acquisition of infection in exposed people. So really what you're seeing is that there's a narrow window right after the onset of symptoms where it will decrease the likelihood of bad outcomes. And I think that's an important message. It, like monoclonal antibodies, has a potential role to play. I also think that once we get drugs, because some of these drugs are looking reasonably good, the ease of administration of 5 days of an oral regimen may sort of relegate the monoclonal antibodies and the convalescent plasma to historical significance. But that's just speculation. I don't know what Stefan or Dave think.
Yeah, and so I think there's a lot of truth to that. I just want to point out that convalescent plasma is beautiful in the sense that when you have a real pandemic situation and lots of people get sick, you don't need to stock it, because the more people get sick, the more convalescent plasma you can actually get. Mm-hmm. You don't need to buy it. You don't need to, you just need to cool it for a few days, because in pandemic it'll go fast. And one of our colleagues never gets tired of calling it the drug or the treatment from the people for the people. So that is that's a colorful picture, but it's isn't it interesting, right? So in a pandemic situation, the more sick people you have, the more treatment you have.
So Stefan, is that an Arturo remark?
Yes, it is. Okay.
Okay, thank you, everyone. There are no more questions or comments.
Okay.
Thanks a lot, Jasmine and Edward.
Yep.
Thank you.
Nice job.
Thank you.
Yeah, very good job.